the DNA. DNA stands for deoxyribonucleic acid. DNA is pretty unusual in that it is about the only common molecule capable of directing its own synthesis. The processes of mitosis and meiosis were discovered in the 1870s and 1890s. It was observed that as cells divided, chromosomes move around in a cell and people began to wonder what their function was. It was determined that chromosomes were made of protein and DNA, about which people knew almost nothing. People began to suspect that chromosomes had something to do with genetics, but couldn't explain what, how. When enough evidence was accumulated to confirm that chromosomes did indeed have something to do with genetics, most people thought that in some way the protein in the chromosomes served as the genetic material. People knew that DNA was also in the chromosomes, but because its structure was unknown and people didn't know much about it, few people thought it was the genetic material. A DNA molecule consists of two long polynucleotide chains composed of 4 types of nucleotide subunits. Each of these chains is known as a DNA chain or a DNA strand. Hydrogen bonds between the base portions of the nucleotides hold the two chains together. Nucleotides are composed of a 5-carbon to which are attached one or more phosphate groups and a nitrogen-containing base. In the case of the nucleotides in DNA, the sugar is deoxyribose attached to a single phosphate group, hence the name, and the base may be either adenine A, cytosine C, guanine G, or thymine T. The nucleotides are covalently linked together in a chain through the sugars and phosphates, which thus form a backbone of alternating sugar-phosphate-sugar-phosphate. Because only the base differs in each of the 4 types of subunits, each polynucleotide chain in DNA is analogous to a necklace, the backbone, strung with 4 types of beads, the 4 bases A, C, G, and T. The way in which the nucleotide subunits are lined together gives a DNA strand a chemical polarity. If we think of each sugar as a block with a protruding knob, the 5-phosphate on one side, and a hole, the 3-hydroxyl on the other, each completed chain, formed by interlocking knobs with holes, will have all of its subunits lined up in the same orientation. Moreover, the 2 ends of the chain will be easily distinguishable, as one has a hole, the 3-hydroxyl, and the other a knob, the 5-phosphate, at its terminus. This polarity in a DNA chain is indicated by referring to one end as the 3 end and the other as the 5 end. The 3-dimensional structure of DNA, the double helix, arises from the chemical and structural features of its 2 polynucleotide chains. Because these 2 chains are held together by hydrogen bonding between the bases on the different strands, all the bases are on the inside of the double helix and the sugar-phosphate backbones are on the outside. In each case, a bulkier 2-ring base is paired with a single ring base, a pyrimidine, A always pairs with T and G with C. This complementary base pairing enables the base pairs to be packed in the energetically most favorable arrangement in the interior of the double helix. In this arrangement, each base pair is of similar width, thus holding the sugar-phosphate backbones an equal distance apart along the DNA molecule. To maximize the efficiency of base pair packing, the two sugar-phosphate backbones wind around each other to form a double helix with one complete turn every 10 base pairs. It was known well before the structure of DNA was determined that genes contain the instructions for producing proteins. The DNA messages must therefore somehow encode proteins. As discussed, the properties of a protein, which are responsible for its biological function, are determined by its three-dimensional structure, and its structure is determined in turn by the linear sequence of the amino acids of which it is composed. The exact correspondence between the four-letter nucleotide alphabet of DNA and the 20-letter amino acid alphabet of proteins, the genetic code, is not obvious from the DNA structure, and it took over a decade after the discovery of the double helix before it was worked out. We described this code in detail in the course of elaborating the process known as gene expression, through which a cell translates the nucleotide sequence of a gene into the amino acid sequence of a protein. The complete set of information in an organism's DNA is called its genome, and it carries the information for all the proteins the organism will ever synthesize. The term genome is also used to describe the DNA that carries this information. The amount of information contained in genomes is staggering. For example, a typical human cell contains 2 meters of DNA. Written out in the four-letter nucleotide alphabet, the nucleotide sequence of a very small human gene occupies a quarter of a page of text, while the complete sequence of nucleotides in the human genome would fill more than 1,000 books the size of this one. In addition to other critical information, it carries the instructions for about 30,000 distinct proteins.